Hi all, I am Dr.Bliss Ramya Joan, working as Assistant Professor in the Department of Biotechnology, Bishop Iber College. Now I am going to present on the topic bacteria. So there are two kingdoms of bacteria, that is eubacteria, that is true bacteria, and another one is archaebacteria. The archaebacteria is nothing but the oldest organisms on earth, live in extreme conditions. So what is bacteria? Bacteria are unicellular, prokaryotic. What is prokaryotic? Prokaryotic cells are much smaller than eukaryotic cells. Both plants and animals are coming under eukaryotic cells and bacteria is coming under prokaryotic cells. So prokaryotic cells have no nucleus and like organelles. So all prokaryotic cells are encased by a cell wall. So what is the characteristic features of bacteria? So some bacteria are aerobic, that means that they require oxygen in order to survive. Other bacteria are anaerobes, meaning that they do not require oxygen to survive. Most bacteria are harmless and offer beneficial functions to living things and humanity. So bacteria contain a singular circular piece of DNA. It has tiny circular pieces of DNA called plasmids. It has ribosomes. So this is the structure of bacteria. It is having the chromosomes and ribosomes. It has some plasmids. It has a cell wall and the cell membrane, and it has a flagella for the locomotion. So the reproduction in bacteria, how the reproduction takes place in bacteria, usually by means of asexually. The reproduction takes place in bacteria. That means by mitosis, usually by means of binary fission. So how this binary fission takes place? By means of the duplication of chromosomes, yes, by the dividing of the bacterial cells into two by the continuous growth of the cell, then the chromosomes are duplicates and it's divided into two cells. So this what the binary fission takes place by means of asexually. So how this bacteria reproduce sexually? By using conjugation. So how this happens? By means of exchange of plasmid DNA from the donor to the recipient cells. So this is how the bacteria becomes antibiotic resistant. So how this happens? So the activation of DNA is getting transferred from donor to the recipient by means of the enzyme endonuclease. It cuts at the origin of site and it transfers the DNA to the donor to the recipient. And how this transfer is taking place? By the transferring of genetic material from the donor to recipient by means of this conjugation process. So bacteria have cell walls made up of peptidoglycan, that is polysaccharide linked with chains of amino acids. So this may be covered with an outer membrane of lipopolysaccharides, that is chain of sugar with an lipid flagella for the locomotion, and it has some pili. Pili means it has a short, thicker outer growths that helps cell to detach to surfaces. So bacteria can be moved by air and water currents and on any surfaces such as clothing, hands or any object. It can transfer from place to place. So there are three bacterial shapes, generally rod shaped are called bacillus, and spear shapes are called coccus, and spiral shapes are called spirillum. So these are all these shapes of bacteria: cocci, spirilla and bacilli. So gram stains, okay, so it is important in medicine because it provides information for the treatment of bacterial disease. So bacterial stain, it either stains either gram positive or gram negative. So in case of gram positive, if we want to detect the gram positive bacteria, it shows purple in color. If we want to detect the gram negative bacteria, it shows pink in color. So gram positive, it tends to respond to penicillin and like antibiotics. Gram negative, it responds to types of antibiotics unrelated to penicillin. So what is the process behind the gram staining? So these are all the dye and some of the reagents which is used for the gram staining processes. The one is crystal violet dye, and the second thing is iodine, and the third is alcohol, and the fourth is saffronin. So in crystal violet dye, when we add to the particular bacteria, if we detect it is then gram positive, it shows purple color. Okay. By adding this iodine, it decolorizes. So after adding this alcohol, if it is a gram negative bacteria, so it is having a lipopolysaccharide in their cell wall. So the saffronin, by adding counter sign, it enters into the cell wall of the gram negative bacteria. So the gram negative bacteria, it shows pink in color. So this is the principle of staining technique. So as we have already discussed about the crystal violet, no, it is a primary stain, it acts as the primary stain, and iodine, it acts as a mordant, it fixes the dye, and alcohol, it acts as a decolorizing agent, and at last we have added the counter stain, it is a safranin. So gram stain is usually performed on a smear preparation that has been heat fixed on the slide. So once it is fixed, the following stains are added. One function of fixation is to secure, to fix the cell to the slide. So this is the difference between the gram positive and the gram negative bacterium. So the gram positive bacterium, it shows crystal violet in color, and gram negative, it shows pink in color. So gram positive, the purple, because why the purple colors? Because of the large amount of the peptidoglycan in the cell wall. Gram negative, it shows pink because the lipopolysaccharide membrane is removed by the alcohol, and the thin peptidoglycan layer allows dye to escape, and the safranin, the counter stain, it enters into the bacteria, and the gram negative bacteria, it shows pink in color. So this is the cell wall for the gram negative envelope. How this antibiotic works towards the bacteria. So work by preventing the cell wall formation, it breaking up the cell membranes or disrupting the chemical processes. So antibiotics, it cannot treat the viral infection, only antibiotics, it treat the bacterial infections. So there are three types of bacteria based on how they obtain energy: heterotrophs, photosynthetic, and hemoautotrophs. So this is one example I have shown you in the slide. This is the anthrax bacterium. So most are heterotrophs. Under heterotrophs, there are three types. One is decomposers, that is, it's feed on and recycle organic material, this kind of bacteria, and pathogens or parasitic diseases causing bacteria. It either attacks cells or secrete toxins. Nitrogen fixing bacteria, that is rhizobium. So it is found in nodules of soya beans, peanuts, alfalfa, and clover. So it converts atmospheric nitrogen, that is N2, into ammonia, called nitrogen fixation. It is used in crop rotation. So decomposers, so bacteria are decomposers, right? They break down the chemical elements inside other living or dead organisms. Some bacteria live in the intestines of humans and animals. So rhizobium bacteria, it converts the nitrogen gas into other substances that help the plants grow. Pathogens, so how this pathogenic bacteria, how it affects our humans in case of strep throat. So how the infection occurs by means of the streptococcus and the staphylococcus, another species of bacteria, how it infects the humans. So this is the example behind the types of bacterium, how the infection caused in humans. Nitrogen fixing bacteria, so how the nitrogen is fixing the root nodules in the rhizobium, okay, nitrogen fixing bacteria on the nodules of roots. Some like cyanobacteria are photosynthetic, so these are autotrophs that use the sun's energy to make foods. So these bacteria are chlorophyll and uses the process of photosynthesis to produce food, very like a plant, very much like a plant. Chemoautotrophs, it obtain energy by removing electrons from inorganic molecules such as ammonia and methane to make food. Examples, Nitrobacter and Nitrosomonas. It lives in soil. It have a crucial role in nitrification. That means it turn ammonia into nitrates, the form of nitrogen commonly used by plants. Some good bacteria I am going to discuss. So lactic acid bacteria and wastewater bacteria. So under this lactic acid bacteria have been used to ferment or culture foods for at least 4,000 years. For example, products like yogurt and cheese are coming under this good bacteria and lactic acid bacteria. Under wastewater bacteria, it feed on everything from solid human waste matter to last night leftovers. As the bacteria eat, they convert organic matter to carbon dioxide, releasing electrons, the basic element of electrical current. So thank you all for the patient listening. Thank you.